Hello and welcome to the DDW sponsored vodcast series, where we examine the people, the breakthroughs and the innovation driving the drug discovery and development sector. I'm Megan Thomas, Multimedia Editor at DDW. Drug Discovery World is the multi-platform global voice for the drug discovery and development community. From research stage to clinical trials, the brand covers the latest innovation and expertise in the sector. Above all, DDW readers learn how to turn science into business. Today's episode is sponsored by the Jackson Laboratory. Today's guests are Michael Ellis and Tim Robertson. Michael is the Senior Director of Research and Development at the Jackson Laboratory. Tim serves as CEO of TLR Ventures and leader of the JAX Digital In Vivo initiative. First, a little bit more about today's guests. Michael Ellis leads an extensive portfolio of new product and technology development, along with process improvement initiatives. His role encompasses both internal projects at JAX and collaborations with a global network of partners. He has been instrumental in bringing innovative products to market, including humanized mice, human antibody models, and software leveraging computer vision. Tim Robertson leverages extensive expertise to drive innovation in preclinical research, particularly in the intersection of artificial intelligence, digital monitoring, and drug development. With over 25 years of experience creating companies from novel ideas, he has successfully delivered transformative solutions that address complex challenges in pharmaceutical research and development. I'll start with a question for you, Michael, which is JAX has a long history of expertise in mouse models and research. Can you tell us a bit about the problem you set out to address and how that legacy shaped the initial vision for Envision?
Sure. Yeah. As you mentioned, JAX does have a long history, actually a 95-year history of driving innovation in the use of animal models in biomedical research. In fact, this innovation is really a central part of our mission as a not-for-profit research organization. You look back into even the 1990s and early aughts, people like Rob Taft here at JAX have recognized challenges in the field, in this case with research reproducibility, and we've invented novel approaches to solving that. In the case of Rob Taft, genetic stability was the novel invention that he brought to the table, and it mitigates genetic drift and ensures that the Black 6J mouse that you received today is genetically identical to the one that your peers received a decade ago. This is industry practice today, standard practice. You look further back in time to the 1960s and 70s, people like Edwin Less saw a problem of animal health standards in the industry and invented here in Bar Harbor the individually ventilated caging that is now standard industry practice. And I think that the Envision platform that we're talking about here today is really another great link in this chain of innovation that JAX has brought to the industry, really continuing to improve upon animal research, both to increase the rate of efficiency of discovery and development, but also to improve the stewardship of the animals that are in our care. In fact, problems that first motivated us to begin development of Envision were really the same ones that Rob and Edwin and others in JAX's history were trying to address. We're really here talking about reproducibility, translatability, and throughput. I don't think it's controversial to say that these challenges, we have these challenges in the life sciences space, and I don't think it's controversial to say that they continue to plague us. In fact, these issues are really deeply intertwined with one another. We often make practical economic decisions in our research and our study designs that end up limiting reproducibility and translatability of our work. It's difficult in animal research to measure things like behavioral endpoints. So we end up creating these artificial constructs that suit the convenience and efficiency of the scientist, but are not really ideal for elucidating the biology that we're trying to explore. And this is really how we end up with assays like the forced swim test, the Y-maze, the rotarod, and all these other assays that really have limited translatability due to the anomalous nature of the way that we run them. And would really be much better, I think we all would agree, be much better to observe these animals in a natural environment, to observe them naturally socially interacting with one another, and extracting from these natural behaviors a biological signal of interest. And what's really worse with these standard assays we use today is that they're really expensive to run. And so we end up taking a very limited number of measurements over the course of a study, often just repeating these data points 2 or 3 times over the course of many weeks, which is completely missing the opportunity to capture continuous data, and in particular to capture data during the dark cycle when these animals are naturally awake and active. And there's lots of very interesting data that we have started to explore and started to share about the benefits of doing research in a dark cycle, and what sort of behaviors we can elucidate there. Really, the end result of all this, though, is that today we have a very subjective measures that we use often in life science research, and we capture it with a very low data density. And so it's really not a surprise that we have reproducibility and translatability challenges. And with Envision, we recognized that computer vision has advanced far enough to be able to enable us to leverage it in scale at a low cost and in a manner that can help us address many of the challenges that we just described in animal research. We've successfully leveraged computer vision in the home cage to continuously and reproducibly capture a wide range of digital measures, from simple activity tracking to respiration and even seizure detection. And I think that as scientists begin to get their hands on this technology and deploy it across their various research programs, I truly believe that we're poised to see a significant shift in the throughput, reproducibility, and translatability of animal research at large.
Great. Thanks, Michael. That was a fantastic answer and really shows just how exciting these next steps and the future in this industry will be. Tim, let's dive into the system you developed. How did you go about solving that problem in ways that others hadn't?
Sure, Megan. Well, first of all, we began by asking the users, the future users of this system, exactly what it was that they wanted. We brought together a group of leading scientists from pharmaceutical companies, biotechs, academics, and leading scientists from the Jackson Laboratory to form a group we called the Digital In Vivo Alliance, which explored very early on where this technology is, where the users would like it to go, and what was possible, because that's often the art in creating a system like this, connecting what's possible with what the needs actually are. We formed several working groups within the Digital In Vivo Alliance focused on various therapeutic areas and areas of interest like neuromotor disease. Michael talked about seizure and the work that we've done there, working on dermatology, working on things like animal health, husbandry, and welfare, the applications of this system across those various applications, looking at respiratory disease, and finding in each of these areas what the key digital measures are that would be useful, that would be valuable for driving translation of results from the preclinical lab to the clinic. And then bringing together machine learning scientists and data scientists alongside those biological scientists to get together craft solutions. So that was really the first step. And there was a co-development process between these biological scientists and machine learning scientists and the greater engineering team within DIVA to create this system. Now, to do this, we had to build a team that was very multidisciplinary and was quite experienced, because as we talked to these scientists, they recognized that where the field is today in artificial intelligence and machine learning is not where it's going to be in a few years. This is a very fast moving area. And so those users wanted a system that would be extensible and future-proofed so that the investments they're making today will continue to pay dividends and expand in the future. And so we built a team of leading Silicon Valley engineers, experienced in hardware, experienced in secure large-scale infrastructure, with experience in machine learning, in application development, and overall product design and development, so that we could deliver this end-to-end solution that would have these long legs that our industry partners and users would want going forward. We built upon foundational models, the latest advances in AI and ML, to develop the computer vision algorithms, the models that would allow us to track these animals, to understand their behavior, to quantify it, to store all that data, and to report it out to the end users. So we've used convolutional neural networks, we've used transformers. These are really some of the latest advances that are driving the incredible changes we're seeing that are being brought about by AI, and we've brought them in for this user group. We've developed a complete user experience where the data capture, the study design, the study monitoring, the data analysis, and even collaboration is built into the application platform so that for researchers in this area, they can get all the information they're looking for at Envision, and they can share that and they can work with it and do the analysis to really understand how their research needs to go. And so we've developed this complete end-to-end system, and it's being continually advanced and developed by this team. So it will grow over time, it will bring in new digital measures over time, and the investments that those users are making today will continue into the future.
Great, thanks Tim for that answer. Michael, following on from that, can you speak to the role that the Digital In Vivo Alliance and your pharmaceutical partners played in providing data sets to train Envision's models and how that impacts the measures you've developed?
Sure. I think that Tim touched a little bit on this when he talked about how we sort of kicked off at the very beginning by first starting with getting input from the ultimate end users. And I think that's anytime you develop a new technology, it's incredibly important to bring in the perspective of those ultimate end users early and often. This is just as true in the life sciences industry that we're in as it is in the tech industry or even the fast food industry, right? You don't get to a Dorito flavored taco shell and something as important as that without a lot of input from the market coming into the organization. And right from the outset, we were determined to bring together a diverse group of organizations into the industry, from the industry, in to steer the development of this technology, and to do things like help establish standards for what it means to validate a machine learning model in this context of use. We didn't really want to bring together just a group of talkers, though. We wanted doers, right? There was a lot to develop here. And the Digital In Vivo Alliance was really born from a group of organizations that shared this vision of the future and that were really willing to roll their sleeves up and get to work making it a reality. Every organization in the Alliance is championing an application of the technology, as Tim sort of described and alluded to. For example, the models for epilepsy or models for neurodegenerative disease or models for neurotoxicity, each of these various applications is being championed by an organization. And as each of those organizations leads these applications, they bring in the rest of the organizations to determine the strategy for the models that we're building, to generate the training data, to annotate it, and even in some cases to actually outright build the models. And this is really key because it means that all of the models that are being built on the Envision platform are trained with large scale, really high quality data sets that were built and curated by really some of the world's experts in that particular application, right? And this is the difference between building sort of generic models that work okay and building really incredibly high quality precision models that perform at peak. And it's really been our design ethos from the beginning, was really built around enabling experts in the world to build tools on this system, as we really knew from the outset that JAX didn't have the expertise in every area of biological research and discovery. We didn't have the expertise in every area to be developing exquisite models in all those areas. For example, JAX is known for mouse models primarily, not rat models, but we are working in rat models. And one of our Alliance members has a lot of deep expertise in that space. And they are bringing to the table that expertise and helping to make sure that the rat models built on the system are working just as well as the mouse models. We can't do that without bringing together folks into this alliance that have expertise in all of these variety of areas. We believe, at the end of the day, we believe that to build the best possible models for every application, you really need to have those experts deeply involved from the outset. And that's really exactly what we've done with Envision and the whole purpose of the Digital In Vivo Alliance.
Thanks for that insight, Michael. And now, Tim, one over to you, which is, how does Envision's scalability meet the needs of both small-scale studies and large multi-site research projects?
We recognized from the outset that scalability would be an important attribute of this system. As Michael described, the history of changes that JAX has driven in the animal research industry, like the ventilated cage, which is now the standard, every cage is ventilated. We believe that this digital transformation is going to follow the same trajectory, and that in the near future, you won't have conventional cages anymore, that every cage will be digital. And so with that in mind, we architected the system for that future. That begins with the hardware, the cameras and other sensors that go inside the vivarium to look at the animals. They're engineered using mass market production technologies. They're not one-off bespoke pieces of equipment. They're more like consumer pieces of electronics that can be built at scale. The data architecture is a very important piece of this. We've generated a secure connection from the vivarium up to the cloud that meets the needs of enterprise IT customers. And with that, we're able to leverage the scale of the cloud to give us effectively infinite computing and storage resource. That means that when we get these very large data streams coming off these cages, we're collecting video 30 frames a second at HD 1080p resolution. We don't have to worry about filling up hard drives. It goes to the cloud. And as the cloud scales, we can deal with that. It also means that we can deploy a tremendous amount of computing power on these sort of cages. We can run sophisticated algorithms because we have the scalability of GPUs in the cloud, and we can run those at the full 30 frame per second rate to pull out very fine details and to get very high accuracy. We're seeing 99% plus accuracy in our ability to detect and identify mice in the cages. And so that means that the system is future-proofed, that as future algorithms come along, we don't have to worry about whether we have enough compute power to do that. We're in the cloud, we can scale up with that. We've also engineered our processes around this for scale. So our development practices, our testing and release processes, how we version algorithms, as these things advance, a scientist will want to know that the algorithm he ran yesterday and published his paper on can be replicated by a scientist a year later, and that she can run those same algorithms. So we've done very careful versioning so that you can know exactly what version was run at what time. And we've brought in global enterprise security and quality standards because many of our customers here are global pharmaceutical companies. And for this system to be widely adopted, it's very important that we meet those requirements.
Yes, absolutely. Thanks, Tim. And now, following on from a lot of the points that you did touch on in your previous answer, as research becomes increasingly digitized, how do you see Envision continuing to evolve to meet the needs of preclinical researchers?
Well, when we began talking to researchers in this area, they often had very specific pieces of the puzzle that they wanted to work on. So a researcher might want to look at a particular sensor and what sort of information that could give. Or the researcher might want to develop an algorithm or a model that looked at a particular aspect of mouse behavior. And the challenge for these researchers is that in order to do the part they cared about, they had to build the entire end-to-end system. They had to do all sorts of things like data management, the security pieces, the user interfaces, the analysis routines, just to do the part that they wanted to. And a lot of that was not really value add for them. What we've done with Envision is create a complete modular end-to-end structure. So we made the first version, the first pipeline that goes all the way from the digital cages to the cloud, analyzing the data, displaying it, and allowing users to collaborate around it. But it's a modular extensible architecture so that users can plug into that, be that other algorithms. So we have a developer environment that users can sign up for. And with that, they can build on top of the core Envision data structures and extend them and focus just on the part that's high value to them. They can access the data in various ways. And we have this digital platform inside the vivarium that they can actually plug into in various ways. So with that extensibility, users can extend it. And of course, the Jackson Laboratory is continuing to make investments to extend this. We continue to have machine learning teams working on this, extending our algorithms, bringing in new digital measures, validating those digital measures, and eventually taking them to a sort of industry standard level of acceptance, be that with health authorities or be that with the scientific community. So whatever a user wants to do with Envision, we've already set the stage for you. And AI is a fast growing field. And with Jack's continued investment in this field, we want to stay continually at the forefront of that. So with that, we've built this not just for the scientists who use it every day to make it easy for them so they can see their data and they can run experiments without knowing what's going on under the hood, but for developers who are comfortable at the command line, who want to write Python, who want to use sophisticated machine learning techniques, they can get under the hood and they can modify and extend things as they see fit.
Great. Thanks, Tim. I have no doubt that that will be very useful for our audience, our listeners, but everyone working in that space. And so I just have one question left, which is, in what ways do you think Envision can fundamentally improve research outcomes, particularly in the areas of drug efficacy and toxicity, which I think is actually relevant for both of you. But Tim, why don't we start with you and then we'll move on to an answer from Michael.
Sure. Well, I believe that preclinical in vivo research is actually the highest leverage point in the drug development pipeline. In the pharmaceutical industry, most drugs fail and most research programs are tremendously expensive. All research programs are tremendously expensive. And those costs go up astronomically when you move from the laboratory to the clinic, when you start bringing humans involved, and the risks and the responsibilities grow appropriately with that. If we can make better decisions at the preclinical stage about what candidates we take into the clinic, that can tremendously have benefits for the pharmaceutical industry and help control those costs and reduce them and get better therapies to patients faster. So that, I believe, is why we're focused on this area. Also, we are in the era of systems biology. The diseases we're looking at today, for the most part, are complex diseases that have multifactorial causes that evolve over long periods of time. And as much as we'd like to be able to simulate those diseases in silico and be able to know if a drug is working by just doing a computer simulation or by using things like organs on a chip or cell-based methods, the reality is we're a long way from being able to do that. And for the foreseeable future, animal models, and particularly the mouse models, are going to be our best way of understanding how therapies work in full living systems. Much of the data we collect in these studies, though, is subjective. It's based on an assessment by a person, often an expert person, based on a scale or some sort of rubric like that, on how well an animal is doing, which tells us how well a drug is working. But no matter how much we train these people, there's still that degree of subjectivity. These observations are also episodic. Typically, they might be a point in time. You might make a measurement once a week, or you might do an assay twice during an experiment. And so you're only capturing a tiny amount of data that can be used in these studies. With Envision, we make these observations continuous. We're collecting 24/7 data in the home cage, and we make them objective. We're using these computer vision algorithms to output these continuous digital measures that tell us exactly, for example, how much an animal is moving, how is it spending its time, which allows us to quantitatively assess the safety and efficacy of a drug. Michael, do you want to build on that?
Yeah, thanks, Tim. I'll say that the pleasure and the curse of working very closely with Tim over these last few years is that he captures these answers so well that there's really not much left to add. But maybe if I could add just one or two things, as Tim talked about sort of the episodic nature of the data sets that we typically capture today. He talked about the subjective nature. Maybe if I could add to that briefly, that the data that we get off of our traditional assays is also confounded by the testing method itself. And when you take an animal out of its sleep cycle, out of a cage, put it onto some artificial test apparatus, run it through for 20 minutes, and then put it back, what you are getting is that animal in an extreme stress state that is very different than its natural stress state. And oftentimes that confounds the results you're looking for. And one of the additional advantages to using computer vision in a home cage environment, in addition to the episodic and subjective aspects that Tim referred to, is the fact that you are making ethological measurements. You're observing these animals in their natural behavior. And that is likely to reduce the confounding of nature of the test methods and be able to elucidate more biology. And in fact, we see this when we run studies on the platform today, where the aspect of continuous data, the aspect of observing these animals ethologically, in particular during the dark cycle when they're active, we see very different results than you would see on these standard test methods. And some of the benefits we get from that are we often see these results days to weeks earlier than you see these behaviors coming out in standard assays. And so there's a time value capture here in research. Tim referred to the millions and billions of dollars that it costs to develop drug. Shaving weeks off of these in vivo studies has real value. And so there's a time aspect of the benefit here, but then there's also very likely, and it remains to be seen, but very likely a translatability benefit by observing these animals in an ethological manner. All this being said, if I could just sort of briefly, at the beginning of this, I talked about JAX as a not-for-profit research institute with a mission to enable the biomedical research community with the best possible tools to discover and elucidate biology. Really, when we talk about the sort of future of biomedical research and how Envision can touch on this and improve research outcomes, at the end of the day, this is still our core mission as an organization, and we will continue to invest as an organization down this path to make sure that computer vision is leveraged as safely, as ethically, and as efficiently as possible to address the reproducibility, the translatability, and the throughput issues that Tim and I have both touched on. And so I would expect going forward you to see a lot more to come from the Jackson Laboratory and investments in this Envision platform to continue to drive that, as well as investments with the rest of the community at the table to help drive standards of how we validate and leverage and use these machine learning-based approaches within biomedical research.
And Megan, you asked about both drug efficacy and toxicity, and I want to talk a little bit about what we're doing in toxicology, because it's an extremely important field. It's a big use of animal models. It's the last set of tests where we determine if a therapy is actually safe before we put it in humans. And with that, there are various very established and developed procedures and processes and expertise for making those assessments. But it's an area where we believe that Envision can have a very significant long-term impact. And there are a few different aspects of that. There are the typical, what they call IND enabling studies, the toxicology studies that are done really for the FDA to make sure that a drug is safe. Everything is done under what's called GLP, good laboratory practice. And we will get there eventually, but that's not where we're starting because the regulatory hurdles there. Where we see us having an immediate impact is in safety pharmacology, where we can assess very early on if a drug is not just effective, but in that same study, is it safe? Are we seeing any side effects that may feed into the drug development process? Can we address those potential side effects and understand them earlier, understand if it's a dose effect or an area under the curve effect, and adjust perhaps the dosing regimen appropriately for that? And so this has the potential to shorten drug development timelines and for researchers to make better decisions early on. I also want to talk about how this relates to cancer therapeutics, which is an area that we're just starting to address within the Digital In Vivo Alliance. You know, we're to the point now with many cancer therapeutics where it's not just a question of does the drug cause an effect on the tumor, a positive effect on reducing the tumor size. Tolerability is becoming an increasingly important measure in these studies and for patient well-being. You know, I'm sure many of us know people in our lives who have gone through chemotherapy of one form or another, and the tolerability of those drugs is a very major concern and a very major determinant of the quality of life for those patients. And that's not something we've traditionally looked at in our animal models. We've simply measured the tumor size and looked, is it growing or shrinking? What we're able to do with this is assess many of those quality of life measures in a more detailed, structured, early way in these models. So, you know, is the drug causing nausea? Is it causing lethargy? Is it causing hyperactivity? Is it causing sleep disruption? All these things that are so important, we can now begin to address those in our preclinical models to get better drugs into the clinic.
Megan, if I can actually add one more thing, you didn't ask about animal welfare in that question. You talked about drug efficacy and toxicity, but I don't want to forget the fact that we are doing animal research here, and it behooves us, and it's our responsibility to do it as ethically as possible, and to take really serious our stewardship of the animals that we're using. And one of the sort of big value propositions of this technology, in addition to everything we talked about in terms of impact of science, is impact animal welfare. And you are really essentially observing these animals 24/7. And in a traditional environment, we might have a human observer observe an animal for 8 to 10 seconds per cage, at most 2 to 3 times a day, but typically only one time a day. And you can only capture so much of what's going on in terms of that animal welfare situation in that very short stint of time. And what we found is that by observing these animals 24/7, leveraging computer vision, and all of the information that we can extract out of that, is that not only can we detect those animal welfare issues much, much earlier in the process, hours to even sometimes days in advance, but we do so with a much higher degree of sensitivity and specificity than we do by a traditional human observer approaches. And so really, we're talking about a technology that has this dual benefit of significantly sort of disrupting how we do science, and much of what Tim just described, and also at the same time improving the stewardship of the animals that are under our care. And I think that's not something to be taken lightly, and something that we don't want to forget here.
Yeah, maybe I'll address one other topic here. With Envision, we built collaboration features into the platform from the start. And that's because the mythical days of a lone scientist working at the frontier of knowledge by herself in the lab, if they ever existed, they're long gone now. Science is a collaborative exercise, be it in a large pharmaceutical company where these teams are often globally distributed, or even in an academic lab where you may have a PI working with multiple postdocs, multiple students to get an experiment done. And we view that collaboration as one of the key pieces of technology that will drive the Envision platform and make it useful and productive. And so we have a sort of social media feed almost within the secure envelope of Envision, where researchers can at each other. I can say, at Megan, look at this. What do you think about this? And you can respond to it. We have the ability for community members who are invited into a project through a secure channel to comment on it, to annotate behaviors. So I might bring in an expert in a particular behavior, have them come into the Envision application, look at the video, look at the digital measures, and comment on those right in the application. And then that information can be fed back to the machine learning researchers to develop more sophisticated algorithms. And we actually see a day not too far in the distant future where when someone publishes an in vivo study, in those supplementary materials will be a link to the Envision version of that study. And reviewers, the community, anyone who's interested can go in and actually access the complete record of that study. And we think that this will be very important for reproducibility, that it will result in better review of studies, and it will make it actually easier in many ways for people to publish their work, because at some level it becomes pushing a button within Envision.
That brings us to the end of this DDW vodcast episode, sponsored by the Jackson Laboratory. If you are enjoying this content, be sure to join Free Today to become a member of Drug Discovery World. Membership includes full access to the website, including free news and exclusive content, unlimited app access, a weekly e-newsletter, podcasts, vodcasts, white papers, ebooks, and information from trusted third parties. Thanks for watching.